So while we're setting up, let me just start off by saying that I'm not going to be half as entertaining as Tim. I'm also not going to touch on anecdotes, anthropology, or agricultural policy. I will touch on associations, but I will only do so in general when you have meta-analyses with robust data. I will mainly want to focus on controlled clinical trials, which offer the best evidence for the points I'm going to make. I was going to touch on the WHI lightly, but now I'll have to say a little more when I get to that part of my talk. It doesn't show anything like what he just told you. But I think all of the other points that he raised, I will touch on during the course of my talk, which is good because I also have a lot of slides. I have a lot of slides because this is going to, be recorded for posterity, so to speak. So I apologize. I'm going to go fast, but I'll try and summarize every segment as I end to get the take-home message. And I guess we can all catch up on whether I said it right at the end when we go back to the website. So I have no conflicts of interest. I'm going to divide my talk into three segments: serum cholesterol as a risk factor, the diet-heart hypothesis, and diet, obesity, and diabetes. Now, that first one. So my framework for this part of the talk is summarized here. So epidemiology and basic science informs the need for efficacy trials, effectiveness trials in the population, the guidelines, implementation research. And the goal here is to bend the curve of an increasing rate of heart disease towards a decreasing rate of heart disease. I actually made this slide from another slide which was for hypertension because the same principles apply for all the risk factors. And so historically, total cholesterol 50 or so years ago was divided into these fractions: VLDL cholesterol, LDL cholesterol, HDL cholesterol, and this was the bad cholesterol, high levels increased the risk, and this was the good cholesterol, high levels decreased risk. But we know now that it's not these fractions, it's the particles that confer risk or protection. So from the diet, you get these chylomicrons and chylomicron remnants. These are triglyceride rich. From the liver, you get VLDL particles, which are also triglyceride rich, and they get metabolized as the triglyceride gets stripped off into intermediate and low density lipoproteins. And these are all bad particles. They all confer risk. And these are the good particles. HDL involved with reverse cholesterol transport is protective. They also come from the liver. So why are the LDL particles bad? Because they migrate into through the intima into the vessel wall. It's a pure mass effect, a concentration gradient. There's no LDL receptor on the vessel wall. The more LDL particles there are, the more get into the vessel. These are modified, oxidized, and that causes endothelial dysfunction, and that in turn causes monocyte adhesion, and the monocyte migrate into the vessel wall, take up these particles. These are the foam cells. Eventually, the lesion ends up with both free cholesterol in the form of crystals and esterified cholesterol, and that's the vulnerable plaque. So HDL particles are good because they inhibit this adhesion molecule expression, so the monocytes don't stick, inhibit the oxidation of LDL, and promote cholesterol efflux. So historically, as Tim has said, we've measured triglycerides, which are mainly in these moieties, LDL cholesterol and HDL cholesterol. Those are the well-known lipid risk factors. But now we're able to measure the actual particles through nuclear magnetic resonance. So we can measure these VLDL particles, the LDL particles, and the HDL particles. But NMR is not available to everyone. So there are some shortcuts we can take. But before I get to that, let me make an important point. And that is that, you can't read this, so that's okay. There are about 50, that the LDL particle number is more important than the LDL particle size. So there have been 50 studies, there have been in 2006, 50 studies looking at the relationship of particle size to coronary heart disease risk. Of those 50, only 12 showed a significant association with coronary heart disease after adjustment for other risk factors. There have been a smaller number of studies looking at LDL particle number, 4, and in these the independent association remained after adjusting for other risk factors. So it's particle number, not particle size. All of these particles are bad, except for HDL. And here's an example of one of those studies that I showed you, the study by Lou Culler. So here's the LDL particle number by NMR. This is adjusted only for age and race. It shows a strong association. Here's the particle size. Shows a significant but weaker association. This goes away after adjustment. This does not. LDL, cholesterol, whether measured by NMR or conventionally, is highly significant and remains significant after adjustment. So it's the particle number. I'm not saying that different particles may not have slightly more effect, but the main thing is the particle number. Now, since we can't always, not everybody has access to measuring particle number, we have to look at alternatives. And one alternative is to measure ApoB. Every one of these particles contains one molecule of ApoB. So if you measure ApoB, you've got all the particles. And for APO, and you can get an idea of the LDL particle number by measuring the major apoprotein, which is ApoA1. Or alternatively, and even more simply, you can measure non-HDL cholesterol, which is in here, and HDL cholesterol. And it turns out that if you do that, you get very similar answers to measuring the apoproteins. To get to that point, I want to point to an even larger meta-analysis than the one that Tim showed you of major lipids, apoproteins, and risk of vascular disease. 302,000 participants from 68 long-term studies, including the famous Framingham, but many others. So this is huge. And they chose to focus on this, on non-HDL cholesterol and the apoproteins for technical reasons, which I won't get into too much. I'll touch on it. But anyway, the meta-analysis showed that the association with heart disease, coronary heart disease, was pretty much the same whether you measured non-HDL-C or ApoB, and the protective association with HDL was this, HDL-C was the same as with ApoA1. So it doesn't matter. You can go the low-tech way and just measure non-HDL cholesterol or HDL cholesterol, and you get the same answer as the more high-tech approaches do. So using this approach, triglycerides appear to be a risk factor for coronary heart disease, but that association disappeared when fully adjusted. HDL was protective, so, and that association remained after adjustment, although attenuated. Non-HDL-C was a strong and independent risk factor with an adjusted hazard ratio of 1.50, highly, highly significant, and didn't change much with adjustment. I'd also point out that at the level here of 130 milligram per deciliter, that corresponds to about 2 millimoles per liter of LDL-C. Very few of us have that level. The point is that the association of LDL with coronary heart disease occurs even at the lower end of the distribution. So it applies through the entire range of LDL. It also applies irrespective of age, of sex, and of a history of diabetes. So LDL remains important in diabetics and in old people. These are people over 70, and in women. So to summarize this part of the talk, as I promised, so LDL cholesterol remains clinically useful. Non-HDL cholesterol is useful because it also has the advantage you can do it in non-fasting subjects. It's a proxy for the atherogenic particle number. The hazard ratios of the non-HDL-C are similar to that of measured LDL-C, which I skipped over in the interest of time, and of ApoB. Remains independent, strong independent risk factor after adjustment. Older people, women, and diabetics. HDL-C remains as an important protective independent factor. Triglyceride is not an independent risk factor in this huge meta-analysis. So now I want to get to the important issue. Okay, so here you have a risk factor, but is it simply a marker or is it causal? And what I'm going to show you is that it's causal. It actually causes heart disease. So it's integral to the initiation and progression of coronary disease. The estimated heritability is 40 to 50%, and amongst that heritability, there are some rarer but very strong monogenic Mendelian disorders, which can cause either a very high LDL cholesterol or a very low LDL cholesterol. So I'm going to point to the example of the LDL receptor mutation, which is familial hypercholesterolemia, causes a very high LDL, and then conversely, the PCSK9 mutation, which causes a very low LDL. And the question is, how does that affect your, over a lifetime, and exposure to those very different levels of LDL cholesterol? How does that influence your risk? And this is like a randomized trial, but it's a genetic randomization and applies over a lifetime. And if you have a randomized trial and you find a strong effect, then that's causal. It's not just a risk factor, it's a causal factor. So just very quickly, I'm not going to go into detail. Here's a cartoon of the LDL receptor in the liver. The receptor binds the ApoB on the LDL, internalized, degrades it. Now, in conditions of low hepatic free cholesterol, such as induced by statins, or a diet low in saturated fat and cholesterol, you get a decreased free cholesterol, and this activates the pathway for synthesis of the receptor. But there's also a counter-regulatory mechanism through this PCSK9, which is a protease, and it degrades the LDL receptor. So the one mutation is one where you don't have LDL receptors or defective LDL receptors, very high cholesterol. The other mutation is of these are loss of function mutations, where you actually have increased LDL receptors because they hang around for a long time. So familial hypercholesterolemia, not going to spend an awful lot of time on that. Familiar to you, but very important to this discussion. This is in Japan, not otherwise known as a high CHD rate country, from Hiroshi Mabuchi, and it shows the early CHD rate in men and in women. The mortality from familial hypercholesterolemia in these Japanese men, the average age of death is 26 years, was 26 years. And in women it was 50 years. Now that's changed because statins help to prolong life in these people. I should also mention that some of the early work on the receptor was done here in this faculty by people like Denise van der Westhuizen and Jerry Coetzee and Wieland Pievers. They did some important work on the Afrikaner mutation for familial hypercholesterolemia, which explains part of the high rate of heart disease in the Afrikaners. And Priscilla here and I were involved in a study in the Southwest Cape when we were at the MRC, showing that the prevalence of those mutations in the Afrikaners was about 1 in 70, whereas in the world it's about 1 in 500, which does explain a lot of the burden. Now, the next study I want to point to is the converse, which is the mutation of the PCSK9. So this is the LDL distribution in people without the mutation. This is the distribution shifted to the left, and those with the mutation, they have a lifelong 28% reduction in LDL cholesterol. And that lifelong 28%, this is in blacks, 28% reduction translates into an 88% reduction in their CHD rate. So their entire risk almost is explained by this one factor, which is a genetic factor, but it's of lifelong exposure. In whites, the mutation is a different mutation. There's only a 15% reduction, but even that modest reduction translates into a 47% reduction in risk. That's causality. The other, so these authors conclude, and let me just mention one more thing, and that is that Jonathan Cohen here was a member of this faculty in the late 70s and early 80s. He now works at the University of Texas Health Sciences. And this was a breakthrough study, very important study. Yeah. Is he here? Yes. Oh, hi, Jonathan. He actually worked with us a little bit at the MRC for a while too. But he was working on postprandial lipids. So you probably like the non-HDLC part of the story, right? So Jonathan and his colleagues conclude that moderate lifelong reduction in plasma level of LDL is associated with substantial reduction in coronary heart disease, even, and this is important, even in populations with a high prevalence of non-lipid cardiovascular risk factors. Shows how terribly important this one thing is. And the other proof of, and then this is not the only mutation that causes causes, not associated with, causes low LDL cholesterol levels. The others that are somewhat weaker, there's the PCSK9. The others that have somewhat, I mean, these are very modest LDL changes or levels, but they're associated with a pretty substantial reduction in CHD risk. Much more substantial. This is an average of those. So those mutations give a 54% reduction in coronary heart disease compared to the 24% reduction in the statin trials. So it's lifelong, it's dose times duration. Now in the trials you get the same dose-response effect. This is a meta-analysis of the trials at the time, and we found that the reduction in LDL cholesterol was directly associated, or the reduction in coronary heart disease risk was directly associated with the degree of lipid lowering. So summarize this part. So Mendelian randomization studies show a very strong reduction in CHD risk, 54% for 1 millimole reduction in LDL. Clinical trials, 24% reduction for 1 millimole over 5 years, and these demonstrate that LDL is causal. This is incidentally not true for triglycerides and HDL cholesterol, where the same kind of Mendelian randomization studies have not shown this causality. And trials have been equivocal about whether lowering triglycerides and LDL have cardiovascular benefit. So LDL is a far more important risk factor than either triglycerides or HDL in terms of causality. And the reduction in LDL cholesterol in risk occurred in people with disease, in people at any age, in men and women, in those with hypertension, and in those with diabetes. So you can use statins in diabetics and they'll do fine. And it's also effective in people who have low baseline risk. So on the top here are people who would not normally be treated according to current guidelines, but they show a reduction in risk, and that reduction in risk outweighs any harm. Some of the harms are more diagnosis of diabetes, myopathy, hemorrhagic strokes. But since there are 11 fewer major vascular events, and you know, you have like 6 adverse events, even if you say that diabetes is as important as major vascular events, which I don't think is true, there's twice more benefit than risk. And in higher risk people with higher baseline risk, there's 5 times more benefit than risk. So now I want to turn to secular trends, community intervention studies. Finnish men had the highest CHD mortality rate in the 60s, but then since then the decline has also been very striking, most rapid in the world, and about 75% of the observed reduction is explained by a decline in blood pressure, cholesterol, and smoking. So, and the biggest change, according to the Finnish investigators, was the change in health behavior, especially in diet. Finland was a country of dairy fat, dairy farming. They ate a lot of eggs, dairy products, and butter and milk production was subsidized. There was no vegetable oil. So they changed that, and this was a good agricultural policy. They changed it so it didn't subsidize the dairy products anymore, started a vegetable oil industry, started a health education program, and the result was this. They had a reduction in total fat, saturated fat, polyunsaturated fat intake, and reduction in dietary cholesterol. The blood cholesterols went down. The body mass index in men went up by 5%, but didn't change in women. The blood pressures went down, smoking rates went down. And here's the decline, 80% reduction over 35 years in CHD risk, most of it attributable to changes in risk factors, and most of that attributable to the decrease in cholesterol. In the US, there was a steep incline in the early part of the last century, and then here's the introduction of the dietary guidelines, and here's turning the curve that I showed you earlier. Now, this is a slide which is similar to the slide that Tim showed you for diseases of the heart, and that includes heart failure. If you look at ischemic heart itself, coronary heart disease, then the curve bends much more sharply, and I'll show you that in a minute. This introduction of this policy and the guidelines was associated with a reduction in population LDL cholesterol levels. In the UK, they got the message a little later, 10 years later. They changed their dietary fat intake, and the result was that they bent the curve 10 years later. Here's the reduction in the US, which, as you see, for coronary heart disease is much sharper than for total heart disease. So, summarize here, cholesterol is important, it's causal, and it's modifiable, which is a good thing. It's associated with an increased risk of coronary heart disease, even at the normal levels of cholesterol. Biology supports causality, as does the success of treating the high levels. That's causality. And population risk decreases with adoption of cholesterol-lowering strategies. So, our friend the diet heart hypothesis we turn to now. So, here's the old version of the diet heart hypothesis. That total fat and saturated fat increases total serum cholesterol, coronary heart disease increases. Now, the most effective way of showing this association, or the most credible way, is through controlled feeding studies in humans. Those are the studies by Keyes and Hegstedt initially. And the most successful way of showing this link between serum total cholesterol and coronary heart disease, I've shown you, it's the epidemiology. Studies that tie the whole chain together are the ecologic studies across populations and animal experiments. I'm not going to touch on animal experiments. Here's another version of these ecological experiments, not the Keyes one. But you'll see that there is a wide variation. So although there's some association, there are obviously other factors in each country that are important. But I'm not going to dwell on that too much. I mean, I would certainly accept that cholesterol isn't the only risk factor, but I would say it's a very important and causal risk factor. But the diet hypothesis has moved on since those days. We now recognize the role of unsaturated fat much more greatly, trans fats, LDL cholesterol rather than total cholesterol. And we now have meta-analyses of controlled feeding experiments to give us in humans to give us more robust data. And we have these meta-analyses to show these links. I've already covered that. I'm not going to go back to that. That's proven. For the whole chain of events, we also now have meta-analyses of cohort studies of food intake, clinical trials of diet, and population intervention studies and secular trends. So very quickly on the feeding experiments. So when you substitute fats for carbohydrates, the saturated fats increase the LDL, monounsaturated decrease, polyunsaturated decrease even more. All fats, when substituted for carbohydrates, decrease triglycerides, and all fats, when substituted for carbohydrates, increase HDL. But as I've said, it's LDL, that's where the action is. And if you tie it together for the ratio of total to HDL, then saturated fats that don't have much effect, this is not significant, whereas monounsaturates decrease the ratio and polyunsaturates decrease the ratio. However, hidden in this is the fact that all saturated fats are not equal. These are the two common ones, palmitic and stearic. Stearic doesn't have much effect. That's the second most common one. The one with the most adverse effect is this one here, palmitic. It's everywhere. So these studies didn't always capture what type of fat was being included. But they did capture the trans fats, which turn out to be very important. They're even worse than saturated fats. These are usually partially hydrogenated fats. So turning here first, so here's the LDL raising effect of saturated fats. The polyunsaturates decrease LDL, the monounsaturates decrease, and the polyunsaturates decrease even more. And they all, as I said, increase HDL, but the trans less so than the others. And so the ratio on the trans fats, if you look at the ratio of total to HDL, it's worst on trans fats. So trans fats are the worst of the worst of the worst. I'll show you later. So Tim has pointed to this study by Siri Tarino, which is an association study of diet intake with coronary heart disease, and it was null. There was no association. Okay? Now, Thierry Serino et al. Point to some very in there. You always got to read the weaknesses in the comment section. They say that the accuracy of the dietary assessments is a weakness. It's actually a major weakness, and I don't have time to go into it. They're inaccurate and imprecise. And then there's underreporting of calories and fats, I would add, in people that leads to, particularly in overweight individuals. And then there's insufficient statistical power to look at the CVD risk when you replace saturated fats with polyunsaturated fat or carbohydrate. But there are fortunately other studies that have done that. And they point to the importance of looking at clinical trials. So I'll do that too. So in another meta-analysis, the risk for CHD, when you change saturates for polyunsaturates, decrease significantly. When you change saturates to carbohydrates, it increases significantly, but that increase is due to a change from saturated fatty acids to high GI carbs. There's no increase in risk when you change it for low GI carbs. And then coming to the trials, so there's the predicted polyunsaturated fats replacing saturated fats. So here's the predicted effect from those equations from the feeding experiments I showed you. And here's what was found. So in the 8 trials that were done doing that, there's a significant benefit, replacing polyunsaturates for saturates. Good. And the pooled analysis observational cohorts is consistent. So this is very consistent. Polyunsaturates are better than saturates. Clear. Carbohydrate replacing saturated fat is less clear. Here's the predicted effect, but as I've shown you, it depends what carbohydrate you're using, and this doesn't capture that. This is all carbohydrates. And here's the infamous or famous Women's Health Initiative. Women's Health Initiative was not a trial of lowering saturated fats to lower coronary heart disease rates. That was not what it was designed to do. It was a trial of reducing total fats and increasing carbohydrates to see if it would reduce breast cancer. It didn't quite reduce breast cancer risk, but got very close. So the WH trial was to reduce total fat. CHD risk was actually significantly reduced in the subsets of women who achieved lowest rates of saturated fat, trans fats, and highest levels of vegetables and fruits. So it's perfectly consistent with the dietary guidelines. If we wanted to design a trial for the dietary hypothesis, it would have been a different trial. That wasn't our trial. And I think the issues that Tim points out are simply typesetting errors, and I wouldn't be not too worried about those. But we did disclose everything in our paper. So the diet heart hypothesis is alive and well. Elevated LDL cholesterol increases risk of CHD. Saturated fats decrease LDL and the risk of CHD. The type of saturated fat matters. Palmitic is the worst. Unfortunately, it's the most common fat. It's everywhere in the meat, dairy, everywhere. And substitution with polyunsaturated fats is a good thing. It lowers LDL and risk. Saturated fat with carbohydrates, the best I can say there, it does not increase CHD risk. I can't tell you it decreases CHD risk, but it does not increase CHD risk. And the carbohydrate quality matters, and substitution should be with should not be with high GI carbohydrates, because that does increase risk. Jimmy, how am I doing, man? I'm worried that you're going to come in. Okay. So diet, let me turn to my next topic, which is diet, obesity, and diabetes. So I'm going to just make a few ex-cathedra statements here, and I'm not even going to try to defend them. But long-term excess, I don't have time, excess intake, energy intake, coupled with reduced activity increases weight in many, not all people because of this homeostat, but in many people. Maybe in most, it increases weight. And the modern lifestyle, when coupled with a physiology which is geared towards energy conservation, which is fat, is conducive to obesity. So it's much easier to avoid that weight gain than to treat it once you've got it. So that's where we should go in prevention. Non-surgical reversal of obesity requires moderate but persistent decreases in energy intake coupled with some increase in physical activity. Energy intake is the important thing. The point I will address extensively is that this decrease in energy intake and the favorable effects can be achieved with a variety of dietary patterns. And that brings us to this issue, which Tim regards as very important, and that is this low-fat, high-carb diet is the ultimate evil. And some of the criticisms in generalization is that they stimulate insulin and worsen glucose tolerance, they raise triglycerides and lower HDL. That part is true, except when you get weight reduction, and they promote weight gain. Not true. So do fats or carbohydrates have unique roles in obesity? So we can't do experiments ethically to make people fat. I mean, they may do it naturally, but you can't sort of randomize people and say, okay, I'm going to make sure you get very fat, and you got to, lay off the fries. But we can. So we've got to turn here to cohort studies and to secular trends in human populations. We can, of course, do experiments to treat or prevent or treat obesity, and so we've got short-term comparisons of calorie-restricted high-fat and high-carb diets. And we've got one long-term RCT of low-fat, high-carbohydrate diet, which is WHI. And we've got surgical calorie restriction data to show that calories count matter a lot. So here's a very nice analysis of 3 Harvard cohorts looking, and this is nice because it looks at food, not nutrients, but foods, the stuff that people actually eat. And the foods that are associated with an increase in body weight over 4 years are shown up here. These are potato chips, potatoes or fries, processed meats, red meats, butter, sweets and desserts, refined grains, and then there are some foods that are associated with a low gain or reduction in body mass. And these are vegetables, nuts, whole grains. Whole grains, that's carbohydrate. Fruits and yogurt. Fruits also contain carbohydrates. Beverages, sweetened beverages and fruit juice, high GI index sugars increase body weight, or associated with the increase in body weight. I've got to be careful here. Whereas whole milk is not. So other lifestyle factors that were associated with changes in body weight are physical activity, alcohol, smoking, sleep, and watching TV. These were all significant. So it's the whole lifestyle, guys. It's the whole lifestyle. It's not whether you eat fats or carbohydrates, but just stay away from those high GI carbohydrates, the potatoes, the chips and the fries, and the meats. And you should be okay. Eat lots more fruits and vegetables and nuts. So whole grains, I just want to point out that whole grains are associated with cardioprotection, and meat and fats are not. This is from a meta-analysis, and this shows very nicely that for both CHD and diabetes, whole grain consumption is associated with a decrease in risk. And as far as meats are concerned, meats are generally associated with an increase in risk, but particularly processed red meats, and they're associated with increased risk for diabetes as well. So for coronary heart disease and diabetes. So if you're going to eat your meat, lean meat, not processed. So the cohort studies of dietary habits and obesity then, summarize that part. The methodology is awfully challenging, but when you have a cohort study where you can look at things that go in different directions, then you get better information because you've got 2 things going in different directions, so your ability to spot whether something real is going on is improved. And well-conducted studies indicate that people who gain weight and therefore are likely to be overeating have a poor dietary quality, as I've pointed out. And meta-analyses show reduced risk of CHD and diabetes associated with whole grains and increased risk with processed meats, which are high in fat and salt. So this is fun. This is an interesting thing. The secular trends in human population. So there are many studies that have been done, and I've just captured a few of them here. So in all of these countries, there's been a dietary education policy or program, and they've reduced their fat and saturated fat increased their polyunsaturates, reduced their cholesterol, reduced their heart disease risk. What happened to their weight? Well, it's very mixed. In Finland, as I've indicated, women didn't increase weight. Men had a small increase. In the USA, it did increase, but not in parallel. There's a disjunction there, which I'll show. In the UK, it increased, and in Poland, women increased, men didn't change their weight. And in Sweden, the weight increased up to 2004. I'll come to after 2004 in a minute. Thank you. So in the in the USA, years when the dietary recommendations were implemented, it was 3 decades later when this explosive growth in overweight occurred for men and for women. So it doesn't take 3 decades for a change in diet to result in obesity. That doesn't make sense. It's something else, and maybe this is part of it. So this is it's an exaggeration. It's real. I mean, you can buy this, but it's an exaggeration. But the change in dietary habits in the US included the supersizing of foods, both in the home and in fast food restaurants. Plates got bigger, increased sedentary living, more TV watching. It's the whole gamish. It's not really what you're eating, it's how much you're eating and what else are you doing. So I want to turn back to Sweden, because I like this. So after 2004, the Swedes fell off the wagon, and they increased their dietary fat, and their serum cholesterol increased, not yet known how that's going to affect the CHD risk, but I bet you it's going to go up. And now here's the kicker. Both during this period of compliance to the guidelines and the subsequent recidivism, body weight just kept on increasing. So it wasn't what they were eating, it's how much they were eating and what else they were doing. Okay, I'll show you more detail. So in the 1970s, they also in Northern Sweden had a high risk. They started a community intervention program, and they called it a modified Mediterranean diet, modified in the sense that total fat was reduced, but they shifted from saturated to polyunsaturated fatty acids, fewer eggs and more vegetable fruit and all the good stuff, whole grain bread. And their rates declined by 50%. So in this process, they increased their carbohydrates, decreased their fats. This is men, this is women. And then in 2004, because of an enthusiasm for the Atkins-type diet, the media went crazy about the low-carb diet. They decreased their carbs, increased their fats in both men and women. Okay? And importantly, is this the 1 minute or the 3 minute? Importantly, whereas before they decreased their consumption of this mix of butter and rapeseed oil and substituted it for low-fat margarine, then they backslid and they got back on the butter, rapeseed, and then they majorly backslid here in 2004 and took off on and decreased their margarine intake. And their serum cholesterol followed exactly the same pattern. Initial decrease, things were going along, and then when they changed in 2004, it increased again, in spite of an increased use of statins. Without statins, this would have been much worse. And their weight just kept on marching on. So as their composition changed drastically, their weight just kept going up. So it isn't composition. I'm going to skip that. So the studies of secular trends show that the trend towards increasing obesity, not uniquely associated with low-fat, higher-carb diets, other factors responsible. These diets do result in a lower cholesterol levels and CHD risk and mortality, as does the statin trials, incidentally. Implementation has been beneficial, and reverting to a higher-fat, lower-carbohydrate diet, as Sweden did, may not reverse a trend towards obesity, but may reverse the cardiovascular benefits. And so we do not want to follow the Swedish model. That's my message to you. And I don't have time to go into this if I want to stay friends with Jimmy here. But I would point out that studies which have compared, many studies that have compared the low-fat and the low-carb approaches find equivalent effects. This particular study by Foster showed the same effects on the two diets. The only difference was that the folks on the low-carb diet had more dry mouth, bad breath, constipation, and their hair fell out. And I don't have time to go into this study, but I do have to come back to the WHI, where we had we ended in the first year with 48% carbs in the control group, 58 in the treatment group, so 10% percentage point difference. And in spite of loading up, of carbo-loading them, they lost weight. They didn't gain weight, they lost weight. And including in the obese people, they lost weight. So the carbs didn't affect their weight, and their profiles, metabolic profiles, if anything, improved. LDL, blood pressure, clotting factor 7 improved slightly, but significantly. No effect on triglycerides, HDL in long term, glucose or insulin. So it didn't affect glucose homeostasis. And finally, I think finally, you can actually treat people with prediabetes, with glucose intolerance, with the AHA or NSEP step 1 diet, which is a 30% fat diet, and prevent them from developing diabetes. So you can prevent diabetes by feeding people carbohydrate as long as it's energy restricted and they lose weight, because that effect is directly associated with how much weight they lost. If they lost weight, they were fine. If they gained weight, they got diabetes. And then just one point, that the calorie intake obviously matters, and it's shown by the gastric bypass studies where you reduce intake enormously, you lose a lot of weight, you cut your diabetes by 75%, you cut your diabetic dyslipidemia by 60%. So in the longer term, a diet low in fat and high in good quality carbohydrates is not associated with weight gain, increased triglycerides, insulin resistance. Any energy restricted diet that reduces weight will be good. And you can actually use low-fat, high-carb diets to reduce weight and prevent diabetes. And obesity is not the primary driver of insulin is not the primary driver of obesity, as shown here by this is the prevalence of glucose intolerance versus the prevalence of obesity. Obesity is much more common than glucose intolerance, which is the opposite of what you affect if insulin resistance was the main driver. And I was wondering, what percentage contribution does insulin resistance make to obesity? And Tim gave the answer. It's 3%. Because one of the studies that he showed said out of 400 people, 12 had insulin resistance, and that's 3%. And they're the ones who came to grief. So the good, the bad, and the ugly. These are good. These are bad. And trans fats are downright ugly. These are the foods we should eat. These are the foods we should avoid. Really my last slide. I think if you're in prevention science, your primary directive, your primary objective is to do no harm. You're trying to prevent well people from getting sick, so you don't want to do any harm. This is from the Hippocratic Oath, sometimes related as primum non nocere, and I would say that the diet recommendations have done a lot more good than harm, and while we can certainly tweak them, throwing them overboard, as my opponent would suggest, could potentially do an awful lot of harm, and we don't want to go there. Thank you.